Welcome back. Today we are going to start another topic of chapter 5, Principles of Inheritance and Variation. So here we will discuss about the dihybrid cross. As we have done in monohybrid cross, what was the monohybrid? When we are studying one character at a time, or we are doing the inheritance of how the inheritance takes place of one character. So here we will talk about the inheritance of two character or two gene, because we know that character is governed by the genes. Okay, so here we will discuss that how many characters we are taking. Two characters, and each character having two traits. So first of all we have taken first character, that is seed shape. So how the seed shape, it can be round or it can be wrinkled. If you will talk about the round, it will be like this, or if we will talk about the wrinkled, it will be like this. Okay, so we have taken the first character, that is color or shape of seed. It can be round or wrinkled. Another one, we have taken the another character, that is seed color. So the color can be what? It can be yellow or it can be green. Okay, and here we will talk that the round character as well as the yellow character are the dominant characters. Okay, so what are the dominant and what are the recessive, which is showing its expression over the recessive one. And here if we will talk about these two traits, these are the recessive one. Okay, so dominant are the round and yellow. That's why we are denoting with the capital letters. Round capital R and a yellow capital Y. In comparison of that, we will show the recessive character, that is wrinkled, with the help of small case of the same letter, that is small r, and here we will take the green color with the small y, because we have taken the dominant character with the capital Y. Okay, so what we are doing in monohybrid cross, we were taking the tall plant as well as dwarf plant, and what we are getting there, capital T and small t. And after the F2 generation, the phenotypic ratio was what? The phenotypic ratio was the 3:1. Okay, so here what we have done, in place of taking only one character, we have taken two characters. So we know that in monohybrid cross the phenotypic ratio was 3:1. Now if we will take, in place of this monohybrid cross, we are taking two characters. What are the two characters which we have taken? See here, the parental one. Parental means parents. What will be the phenotype of parent? Phenotype is what? The external appearance. It will show like a round yellow. And another one, we have taken parent wrinkled green. Okay, and the genotypes which we have shown of this phenotype, that is capital R and capital YY. Another one, in wrinkled, we have taken small rr and small yy. Okay, now what we are doing here, we are crossing these two characters or these two plants. Now what will happen here, we will get the capital R and small, capital R small r and capital Y small y. So how we are getting here? We know that before this fertilization, what happens? Gamete formation, as we have discussed earlier also. So what will happen? Gamete formation. So if we are having this part, this character and this character, what it will give? It will give two allele, that is capital R and small y from here, and again it will give small r from here and small y from this pair. Okay, so in gamete formation, what is happening? We are forming the gametes, as in case of male gametogenesis as well as female gametogenesis. What happens there? There will be a formation of male gamete as well as female gamete. So here also there is formation of gametes. Now these two gametes will get fertilized, and during the fertilization, what will happen? We will get capital R small r, capital Y small y. Okay, now we know that in monohybrid cross also, what we were doing, we were self-crossing, or we are selfing the F1 generation. So what is the meaning of selfing? When the plants are the same. So the genotype is capital R small r, capital Y small y. So what it is showing? It is showing the round yellow. Okay, why? Because R is dominating here and Y is also dominating. So what will be the round yellow. So what we can say, this is the phenotype of F1 generation, which are all round and yellow type. Now we have done the selfing. So during the selfing also, what will happen? It will give some offspring. So offspring is what? It is the result of fertilization. So but before the fertilization, here also there will be gamete formation, and we have done in the law of segregation. What happens? In law of segregation, the gametes get segregated, but they will not blend with each other, but they will not mix with each other. So what will happen? They will separate to form a gamete. Okay, so how this gamete formation takes place? If I will show you here, capital R, small r, and capital Y, small y. So there may be chances that this capital R can be formed with this Y, and this capital R can form this Y. So again this small r can form with this Y, and this capital small r with small y. So how many gametes we are forming here? 4 types of gametes. Okay, so whenever there will be gamete formation in F1 generation, it will give rise to 4 gametes. Fine. Now I will show you one formula is there. What it is? 2 raised to the power of n. Okay, so n denotes what? It denotes that number of heterozygote. And what is heterozygote? Which are different one. For example, if you will see here, R, small r, capital Y, small y. So what you are watching here, that we are having 2 heterozygote. What are these 2 heterozygote? Capital R, small r, and capital Y, small y. Okay. So if you will see here, 2 raised to the power, how many heterozygotes we are having? We are having 2. So how many gametes it will form? It will form 4 gametes. Okay, so this is the formula to show the number of gamete formation. This formula will be useful to find out the numericals in the later section. So here we should know that how many numbers of gametes are forming here and how they are forming. So another thing which I want to explain it here, these gametes, if they are forming, how we can show in a very simpler form. For example, same thing, we have taken capital R, small r, capital Y, small y. Okay, so what we are going to do here, we are taking the first 3 things, first 2, capital R and small r. Okay, like this you will write it. So first of all, what you will do? You will write the capital R, small r. Now the remaining capital Y and small y we are having. So what is the use of this thing? I am rubbing it here. So the left over part of capital Y, small y, we will write like this. Again we are having capital Y and small y. If you are having any problem to form the gametes like this here, so you can go for this method. This method is a simpler one. So what we have done, we have taken capital R, small r, and the remaining one which we are having capital Y, small y, we have distributed here. Now if we will join this thing, okay, now if we will join this thing, capital R with the capital Y, what we are getting? Capital R, capital Y. Now when we are when we are combining this capital R with small y, what we will get here? Capital R, small y. Now another thing, small r, when it will combine with capital Y, what it will form? Small r, capital Y. Again, in the fourth condition, when this small r will combine with small y, so it will form like this. Okay, so these are 4 types of gametes which we are getting in the F1 generation. Okay, so now which we have formed in the F2 generation. In F1 generation we have this thing, but in F2 generation, after the gamete formation, what we will get? We will get the different types of pairs. Okay, so here we have written the same thing, capital R, capital Y. Okay, capital R, small y from here, small r, capital Y from this side, and small r, small y from this side. Okay, so as we know that both are the same thing, so same thing we will get from this side also. Okay, so these are, we can assume that this is a male gamete and we can assume that it's a female gamete, or vice versa. It's your choice. Or we know that these are pollen grains. If we'll talk about the plants, so male gametes are the pollen grains and the female one is the ovum. Okay, so now what we will do in F2 generation, after the gamete formation, what will happen? They will produce 16 types of combination. Okay, how many? 16, and we will show it with the help of Punnett square method. In Punnett square method, what we are doing? We are arranging the gametes in the upper part as well as the lower part, and we know that the straight line or in the upper part, what we are doing? We are taking the male gametes, as well as the downwards we will take the female gametes. Fine. So what we have, the gamete which we have formed here, we will arrange them in the upper part. So we have arranged capital R, same thing which we have written here, we have written in the columns here. Now, and same thing which we have written in the downward side. Now we will make the combination. How we will make the combination? When this capital R will cross with this capital R, what it will form? Capital R, and here capital Y and capital Y. Okay. Now like this, what we will do? Capital R, capital Y and small y. And again capital R, small r, capital Y and capital Y. Like this, capital R, small r, capital Y and small y. Okay, now like this we will do capital R, capital Y, small y. Then capital R, small y, capital R, small r, capital Y, small y. Okay, like this, capital R, small y, small y. Now the third times, capital R, small r, capital Y, R, small r, capital Y, small y, R and Y, then R, Y and Y. Here also what we are doing? This capital R, small r, capital Y, small y, then capital R, small y, R, capital Y, small y, then R, Y. So these are the 16 crosses which will form in the F2 generation. Now what is our objective? To find out the ratio here. As we have done in the monohybrid cross, what was the phenotypic ratio? It was the 3:1. In mono, when we are taking one character, it was 3:1. When we will take the two character, it will be just double. Okay, 3:1, like this, whole square. So if we will split this thing, what we will get? We will get 9:3:1 ratio. So now we will talk that how we are getting this ratio here. See, in F2 generation, what would be the phenotypes? There would be 4 types of phenotypes. Okay, what was it? Round yellow, round green, wrinkled yellow and wrinkled green. See students, this dihybrid cross, or the basis of this, it is a law of independent assortment. After this topic, we will do the law of independent assortment, which says that what happens, these alleles will get segregated and they will rearrange themselves in the F2 generation. Okay, segregation is there in gamete formation, but in F2 generation all these will get rearranged themselves. So due to this rearrangement, what happens? They will take some character from the parents, as well as they will make some recombinants also. So what are the recombinants? The recombinants are those things which are forming by the taking character from both the parents. Okay, so if you will see the F2 generation, you can see we are having round yellow. Round yellow was the parental type. Okay, as well as you can see the wrinkled green, they are also from the parental type. Now along with this parental character, we are getting some recombinants also. And what are the recombinants? These are the round green and wrinkled yellow are the recombinants. We can see that round can combine with this green and this wrinkled can combine with this yellow. So due to this, we are getting some pairs of the round green as well as of wrinkled yellow. So come to this Punnett square, the thing which we have shown here from this blue mark, you can see round yellow. What this is showing here? Both are dominant character and both are dominant character. So it is showing round yellow. If you will see here, again it is a round yellow. If you will see here, again it is a round yellow. Why? Because on this small r, this r is dominant one, and here in this one, this if you will see here, small r and capital R, which one is dominant one? Capital R. If you will see this capital Y and small y, which one is dominant? Capital Y. So all these here which I have marked with blue marker, it is showing round yellow. So how many round yellow we are getting out of this 16 combination? We are getting, from here I will count 1, 2, 3, 4, 5, 6, 7, 8, 9. So how many round yellow we are getting here? 9. And if you will see the round green, what are the round green? Round is showing with the capital R and green with the small y. Okay, so here if you will see here, what is the meaning of capital R, capital Y? It shows that R can combine with capital R or it can combine with small r. Here also capital Y can combine with capital Y or it can be combined with the small y. Both will show which type of phenotype? Round yellow, which is happening here. Okay, now if we will talk about the round green, for happening the round green, R should be capital, and this place can be of capital R also, it can be place of small r also. Okay, and here for showing the green character, it should be in this small y and small y. It means homozygote condition. So if you will see this condition here in out of these 16, so where you can see here round green can be this type. Okay, and round green can be, if now if you will search, the round green can be this type, and again round green of this type. Okay, so we have done 9 of round yellow and 3 of round green. Now come to the wrinkled yellow. Wrinkled yellow, recessive character should be of the small case. It means that R should be of, both R should be of small case, and this Y can be combined with capital Y also as well as with the small y also. So if you will compare this character here, so how can you say this one is, I am showing with the different one, different color. So this one can be what? Wrinkled yellow, as well as if you will see wrinkled yellow, here also wrinkled yellow. So how many wrinkled yellow are there? 3 wrinkled yellow are there. Okay. Again, now the fourth one, which is like of parental genotype, that is wrinkled green. So you can see we are getting only one, I have shown with the green marker, only one wrinkled green. Fine. So we can say that the dihybrid cross in which F2 generation we are getting 16 combination, and out of 16 combination we are getting 4 types of phenotypes, which is in ratio of 9:3:1. So what would be the phenotypic ratio here in the dihybrid cross? It will be 9:3:1. Okay, and it is a result of what? It is a result of the square of monohybrid cross. Okay, so here we have done how the two characters are inheriting in the dihybrid cross. Now we will study that what is the law of independent assortment. In law of independent assortment, what happens as a result that if we are taking two characters. Okay, there is one thing in this dihybrid cross. If we are talking about two character, so we know that these are placed on the chromosome. Okay, so these are placed on the chromosome. So these characters should not be located at the same place or should not be placed at very near to each other. If one character is at one chromosome, then another one should be in the different chromosome, or if it is present in the same chromosome, it should be placed distant to each other. Okay, otherwise what will happen? There will be chance of linkage. Fine. So we can say that in dihybrid cross, it is the prerequisite that these character or these trait must be placed at a different position in a chromosome, either in a same chromosome or in a different chromosome. So that's, here we have done the dihybrid cross or law of inheritance of two genes. Fine. So let's start with the another one. That is law of independent assortment. So now the law of independent assortment. So the basis of this law is what? Dihybrid cross. So we can understand this law only in the dihybrid cross, which we have discussed before that. So here in this law of independent assortment, we have taken same thing. We have taken the two character, or we have taken the dihybrid cross. So here some statements are given here, some postulates are there, that first one, when the two pairs of traits are combined in a hybrid. Okay, what are the hybrids? Hybrids are the two different characters. With a combination two different character, it is forming some recombinant. So when two pair of traits are combined in a hybrid, the segregation of one pair of alleles is independent of each other pair of allele. So as we have done in the dihybrid cross, we have taken round yellow and wrinkled green. Okay, so example, so the first one, what happens? These two pair of traits, what are the two pair of traits? These two, what are the two pairs which we have taken? Seed color and seed shape. So these two traits, what it will do? It will segregate with each other, and what it will form? It will form some gamete here, and again in the further generation they will assort independently. It means that as we have done the dihybrid cross, they will not move with each other, they will assort independently in the subsequent generation. So here this example, what happens? With the help of this gamete formation, it will form another hybrid, and what it is? When we have combined two traits, what we are getting? We are getting this hybrid, and this hybrid will, what it will do? It will segregate and assort independently. Fine, in the subsequent generation. Second thing about the law of independent assortment, when we are getting F2 generation, okay, in F2 generation we were getting the 16 combination. So these written here, when two independent events occur simultaneously. What is the meaning of two independent events? It means that when we are combining two different traits. If monohybrid, we are taking one trait, and here we have combined these two monohybrids. So here when we are taking two independent events occur simultaneously, then their combined probability is equal to the product of their individual probability. Same thing as we have discussed here, in monohybrid cross we are having ratio 3:1, but when we are taking two different events, it means that first monohybrid one character, second one second character. When we are combining both these things, okay, so when we are combining these two events, or when they are happening simultaneously, what will happen? The combined probability, it means that combined one, that after mixing these two, what we are getting? These are the product of the individual probability. It means that it is a product of 3:1 and 3:1, like this. Okay, so same thing I have shown here. Now in F2 generation, in dihybrid cross, we were getting four types of phenotypes, as I have shown here: round yellow, round green, wrinkled yellow and wrinkled green. So here how many round yellow and how many these we are getting here? In the ratio of 9:3:3:1. So how we have done this thing? If I will show you here, round yellow 3:3. Why? What we have done? If we will take this monohybrid or this part, so we are getting 3:1. When we are taking this one, 3:1. So here we are in first part we are having ratio 3. What it will do? It will multiply with the 3 and we are getting 9. Again there is a possibility for a simpler form. If I will show you like this with another color. Now what happens? 3:1, 3:1. Maybe this 3 can be combined with this 3. So 3:3. Now there is a possibility that this 3 can be combined with this another one, 1. 3:1. This one can combine with this 3, 1:3, and this one can combine with 1:1. So same thing I have written here: 3:3, 9, 3:1, 3, 1 3s are 3, and 1 1s are 1. So here, it is a cross check that how many F2 we are getting here. How many? What would be the phenotype? Ratio 9:3:3:1. And same thing it is written in this statement, that the output or the final result which we are getting, it is a multiplication of both the monohybrid or both the different events which is happening together. Okay, so this law of independent assortment based on the dihybrid cross, and there is one possibility, or we can say there is one exception. Okay, so which we have to memorize, that is linkage. What is the exception of law of independent assortment? Linkage. If I will recall the exception of law of dominance, it was the incomplete dominance. Okay, and if we will talk about the law of segregation, there was no exception. That's why we have said this is the universal one. But here again in the law of independent assortment, the exception is linkage. If these characters will present on the same chromosome very nearer to each other, okay, if these two traits which we have taken, like seed color and seed shape, if they are placed with each other, so there may be chances of linkage or crossing over. Due to that, the result may get changed here. So here there is one criteria or one prerequisite, that these traits should be placed in the different chromosome, as well as if they are present in the same chromosome, it should be distant to each other. Okay, if one gene is there, so another gene must be this side. Fine. So this is the example. So here today we have completed the law of independent assortment also. In the later session we will come to know about the numericals based on these laws. That's all for today. Thank you.